We're going to start now because 5 minutes past the hour, I think, past the 8:30 hour, I think it's as good as any to start. Just want to explain, because there are many, many new friends in the group today. I thought I should just explain the space a little bit. This is actually a private space. The reason why I have to explain this is because some of the friends very sweetly asked whether they'll get CPD hours from this or CPE hours from this. No, I'm not an institution that gives out CPE hours. This is just a very friendly little space, community space that we host every Friday for almost 3.5 years now. That, you know, the whole idea being we want to have a space for uplifting conversations. What we see and what we read and what we watch is always so filled with negativity, and we thought that it's important to have a space that allow, you know, wonderful, uplifting, positive conversations, as well as about learning, you know, learning new things from each other. And so over this 3.5 years, we have wonderful, wonderful speakers. From people have dialed in from Washington, DC, people have spoken from Australia. Of course, lots and lots of Malaysians have spoken on all sorts of things, from endangered species to a Nobel Prize winner speaking on disarmament. So we have been really blessed. And today is super special. Super special for two reasons. One is, of course, this is so topical, right? I mean, everyone on the street wants to know, because we are all lay people, we are not professionals, we do not know the difference between this vaccine, that vaccine, you know, and really there's so much myth going around about vaccinations and whether we should go with it or not go with it. So this is very topical. So I'm sure for that reason alone, there are many, many attendees. But more special for me, to be honest, is because this wonderful speaker is my old friend from school. We have known each other for 45 years, and that's telling. We're actually only 46 years old. we have known each other for 45 years and she comes from Ipoh. So I'm going to introduce her now, right? And I need to look at a paper, even though I know her so well, but I do want to do her justice. So she is Professor Dr. Mathee, all right? And I mean, if you can think about every possible degree she has, all right? She's, of course, a PhD holder as well. From Ipoh, that she was here, you know, and then she studied in KL. She then moved to US. And so now, today, friends, very special. She's dialing in at 7:30 or 7:38 in the morning from Miami. So sweet of her to help to grace this space with us. And what else? She is, let me just admit some friends. Okay, so she has done so much work. She has done a lot of speaking engagements. She has won numerous awards. And on top of all this, you know, she's recognized by so many in so many spaces and in publications and literature. On top of all that, you know what really touches me is the fact that she still continues to spend time in Ipoh with her alma mater that she comes from. It's a small Tamil school in Ipoh, and she still comes back whenever she can to support them, you know, whether it's through money, fundraising, or to empower the students and all that. So she has been doing amazing work. So I think with that, I've spent about 5 minutes describing her. I'm going to now invite dear Professor Dr. Kalai to speak. Over to you, dear.
Okay, thank you, Yadmi. Thank you for the amazing introduction, and thank you for being my friend. Lots of good memories with you. And thank you all of you for having me, and look forward to a meaningful presentation and a discussion that hopefully we all will leave this today with knowing a little bit more and busting some myths in the process. So the first thing is I want to, before I start, is I have a disclaimer. I want to quote a Tamil proverb that says, "Katrathu kaiman alavu, kalladhathu ulagalavu." It's basically, you know, what you know, what you've learned is just a handful of what you call, if you take a sand, that's how much you know, but what you don't know is as big as the globe. or you can say what humans have discovered is but a drop in the ocean of knowledge. A translation was given to me by my husband, which was very nice. So perhaps today I know a little more than you do. Some may know more than I, but together we will enrich each other. That's the goal. You'll never know everything. Uh, so the quick outline is I'm going to sort of, you can't talk about vaccination without talking about smallpox. So I will introduce a couple of terminologies, the types of vaccines, what's the difference between preclinical and clinical research, a quick introduction to the virus and disease. Then we'll talk about the various vaccines in production. And then we talk about the 4 vaccines that have been sort of mostly approved, where we could actually find some data, and we'll get into quickly why, if vaccines are so good, why we don't have that many vaccines out there for all the disease. Most of my data comes out of these FDA documents for Moderna and Moderna and Pfizer, which are about 50-page documents each. And then just literally 2 days ago, Sputnik released their paper, came out in Lancet. And then CanSino, there's some little bit I can tell about CanSino, and definitely a bit more about AstraZeneca, which is also known as the Covishield. So most of you probably had your smallpox vaccine, and maybe the elder generation, me and some of your parents would know that smallpox is a devastating and disfiguring disease. And if you get smallpox, the fatality 30 to 40%. In other words, 4 in 10 people will die. The infection is by contact or inhalation, and it's caused by a virus which is called DNA virus. There are 2 kinds, variola and variola minor and major. Unfortunately, there's no cure. You can only prevent the smallpox. So it was very well documented about the smallpox in history. And one of the early documentation of vaccination, a form of vaccination we call variolation. And basically what they did is in China that they collected the dry scabs from people with smallpox and put it and blow it into the nostrils of the other individuals. And that protected these individuals from getting smallpox. And this process is known as variolations. And then there is in 1500, there is writing shows that in India, they would scratch the skin and introduce the scabs to in the healthy individuals. And just like all these ministers and the presidents who are getting in public to convince the people to take the vaccine, Royal Emperor Kang in 1661 had openly took variolation to convince the people to say that variolation will save your life. And then documentation that variolation was ongoing in Africa came from the fact that when the slaves were brought in 1700s, a doctor in Boston noticed this scarified skin, and then when he asked, the slaves explained to them about variolation. But the most famous story that brought variolation to the Western world is happened in Turkey. Lady Montagu, who is the wife of the ambassador, UK ambassador to Turkey, her son comes down with smallpox, and she heard from her husband comes down with the smallpox. She heard from the servants that they do variolation in Turkey. So she variolates her son in Turkey and protects him from smallpox from a husband. And then she takes the story to a UK, and then she, has an open variolation with her daughter. And then she convinces a doctor to variolate the people there. And, they had 2 to 3% fatality with variolation versus 20 to 30% with fatality without variolation. And then in US, of course, in the Charleston, South Carolina in 1738. Moving forward, as you know, most of you know, when you talk about vaccine, most of you hear about Edward Jenner. He was born, and then of course 1796 he discovered vaccination, and 1840 there was free vaccination all over UK to the children, for the children. So what did Edward Jenner found out? He found out that milkmaids with who had previously had a cowpox were immune to smallpox. So he thought that there must be something in the cowpox infections that protect them. So he took this cowpox source, took the fluid and introduced into a boy, James Pepys, 8-year-old boy, and then he then challenged this child with smallpox and cowpox, and the child was protected. So his colleague, Richard Dunning, said you should call, instead of calling it variolation, you should call it vaccination, vacca for Latin for cow. And you know, when he tried to publish this, he was ridiculed. The church called him ungodly, it's repulsive, right? And the Royal Society, which was the only form of journal you can publish those days, they rejected the paper and warned him not to publish anywhere. But of course, he published it in 1798 with additional experiments, you know, use his money. And as I told you that in 1840, there's free vaccination for all infants. And this Edward Jenner Institute for Vaccine Research is in Oxford. That's where the AstraZeneca vaccine was developed. So since then, you know, 1798 was where smallpox vaccine was introduced. Almost 100 years later, the rabies vaccine came along, and then many more since then. So we will not go through all of them, but there's more than just this. I'm sure you're aware of BCG vaccine that was discovered in 1927 by Albert and Calmet and Camille Query. So let's talk about very quickly what's a good vaccine. A good vaccine should induce an immune response in humans without the disease. What is it, a good immune response? It should generate antibodies. Antibodies means these are proteins that float in your blood that as soon as the virus enters or any pathogen, any foreign things enters, they recognize as, hey, you shouldn't be here. So we should cover it up and direct it for death, right? Degradation and death and degradation. So these are the antibodies that neutralize. Think of it as something to neutralize. You know, if you have a enemy, you want to neutralize the enemy, so you neutralize the pathogen. It should be non-toxic. It shouldn't make you sick. And hopefully you have a lifelong immunity. And of course, many of the major vaccines are discovered in 1920s and thereafter. And what happens is we're now finding out the older people, the immunity is waning. So we recommend the booster vaccine as people age. And there is another subject matter. I'm very much a pro adult vaccination. We can talk about that another time. And so, you know, a good vaccine should be something, as soon as it enters the body, the host immune system, the system that protects you from any illness and infection, recognizes. What would it recognize first? It will recognize something on the surface, right? So it's a good immunogen.
Somewhere.
So most of the good immunogens are the proteins, and many pathogens, right, they also cover themselves with some sugar molecules like polysaccharides, and to protect themselves against our immune system. So what we can do is we can use them, but they're not great vaccine, but you can combine it with another component that make it better vaccines. And then you can also have vaccines that are based on things that are produced by the virus or the bacteria mostly. And they produce chemicals, or we call the toxins, like for example, cholera toxin causes cholera. So you can have a vaccine that based on the cholera toxin, but you basically boil the toxin and give the boiled protein so that it won't make you sick, but it will be there to tell the immune system will still generate immune response. So this is a very active slides, but I just for you to understand is that our immune system have what you call the innate immune system. If you think in a big picture, our skin is the first line of defense, right? So this is our innate immune system. And this immune system is and also we have soldiers in our blood, our lymphatic system, going around the system and making sure that we don't have any foreigners entering our body. And so these are what we call the police or the soldiers. And these soldiers, as soon as something enters the body, for example, if you inject a vaccine, they immediately take it up. And then they take it to their station. And the stations are the lymph nodes, where they present these foreign bodies to our immune system and say, hey, look, we are being attacked. We need to get our cavalry set up to go back and attack this pathogen wherever at the site of the infection. So this is what we call the adaptive. So our immune system adapts and creates all these soldiers. One of the major goal of this adaptive immune system is to create these plasma cells, and which creates these antibodies, this antibody that will neutralize. And in addition to that, they also create what we call memory cells. These cells stay where it is, which can be activated much quicker when this pathogen comes again. So generating memory cells is very important for our immune system to adapt. And if you remember, we always need to get 2 boosters, right? At least most of the vaccines, we need a booster vaccine. Why? Because when you first give the vaccine or when you have the first exposure, you have a very short, a small peak of response. And this usually takes to 10 to 14 days. And this is, there's a very specific antibody is made. Let's not go into that. But about 21 days, usually they will give you the second vaccine, or in some cases, 28 days. And this time, the response is much faster, much stronger. In addition, you also generate what we call the memory cells. So that's the reason we want to have this memory cells. And the second exposure, that is your second shot of the vaccine. So there are about 5 different kinds of vaccine. And the early vaccines were the live attenuated vaccines. Basically, you grow the virus many times until it no longer causes disease, but it will grow and the body will recognize it as a foreign. The one you should be familiar with is the polio vaccine is similar. And all these measles, mumps, rubella, chickenpox, yellow fever, they all are attenuated viruses. That means they are live virus that will replicate. So these guys, you only need 1 shot because they replicate long enough to have the vaccine going. And then you can also have like the BCG, it's an attenuated bacteria. Attenuated means that you attenuate or you stop them from causing disease, but still grows as a normal organism. And then they said, okay, there were some people who were getting sick with attenuated organisms, that they decided, let's try some inactivated vaccine. In other words, you inactivate the entire virus or the entire bacteria using some chemicals. Think of it like boiling your egg, right? When you boil your egg, it becomes like a mass of protein that you cannot go backwards, right? So similarly, you inactivate all this and then you introduce as vaccine. And then they said, you know what, sometimes inactivated vaccine have too many things. Let's see if we can just use a small part of the bacteria or the virus that actually responsible for the disease, right? Remember, when an organism enters your body, it needs to grow, it needs to eat, it needs to grow. So it will bring all the genes it requires to do that, but only one part of the whole system will actually cause disease, like the toxins. So they developed a subunit vaccine, and you can see there are, for hepatitis, papillomavirus, and shingles, there are subunit vaccines, and the toxins I told earlier. And then I told you about the polysaccharide, the sugar molecule, and this is very important because there's only vaccine for meningitis, which can kill you or can cause serious damage, such as quadriplegic or become a paraplegic. You need to have this vaccine, especially traveling in the area of meningitis, what we call the meningus belt. And fast forward, we are in 21st century, two new ways to make vaccines, right? So there's a recombinant vector vaccine that was approved, emergency approval was given in December 2019 for Ebola. As you all have heard, Ebola was quite devastating, and if you get it, you die, right? So they now have a vaccine. And then, of course, much talk about the mRNA vaccine, and that is the SARS-CoV-2 vaccine, and we're going to talk a little bit more about it. So when we know there is a disease, we have, and what does it take knowing that a particular virus or bacteria or a parasite that cause disease to get to the population that's you and me? So first of all, you do the discovery phase, and then there is a preclinical testing. These are all laboratory experiments. And then you have to do the clinical research. So this is where the human trial starts. And then once the clinical research is done, you do a data analysis. And of course, in this country, you apply for a FDA approval. And then only then, once the FDA approves and the data is analyzed, and then they're allowed to apply for license, and then start the large scale production. And then they follow up, and literally phase 4 trials sort of starts here. And if you think about it, and this usual timeline from going to preclinical to all the way to launching, you can see it takes many years for exploratory, and sometimes 10 years, 20 years. But even if you do it within a year, to get to the vaccine, it takes up to 10 years and billions of dollars. But what changed with the SARS-CoV-2 infections or COVID-19 is that in the past, you could only do sequentially because you don't have the money and you need to wait for the money, apply for the grants, and you continue as sequentially. With COVID-19, many of these processes were combined, and that accelerated the whole process of vaccine development. So like I saw it earlier, the laboratory test that you isolate the virus, you sequence, you understand how the humans are responding to it, the immune response. We were also blessed by the fact that we had a huge explosion in computer science, especially in the area of bioinformatic analysis. The computer scientists now started realizing that there's a lot of things that can be done in the biology arena where the information is being generated in huge amount, right, in terabytes. So they developed tools to do that, and they also developed high performance computing systems, and that allowed us also to quickly do a lot of research on the computer and then move to laboratory tests. And here you do express the vaccine, you check the stability, you figure out how to formulate, and then you do animal tasks. This usually takes 2 to 3 months. And then you also do challenge experiment. You give the vaccine, like just Edward Jenner did, you give the virus back and see if it protects those animals. So again, I want to say in the past, it's sequentially with COVID simultaneously. And you keep hearing about human trials, right? You say, oh, it's in phase 2, it's a phase 3. What are those phases? So the phase 1 is basically you only use healthy people, and you use about 20 to 80 participants, of course, after approved for testing. And this checks whether the product you made, the vaccine you made, it's safe in healthy people, right? In the phase 2, you use both healthy and sick people this time. And then you ask the question, how much should I give? Right? You're looking at the safety, and you want to figure out how much to give, right? And then the phase 3, which is very important, not only we're interested in safety, but how efficacious is your vaccine. And it's this time you do double blind, which means that patient doesn't know what they're getting. Even the doctors don't know what they're being given. And then in this group, the phase 3, you have to have placebo. That means you have to control the vaccine people with the non-vaccine people. You must have what we call placebo, which is no vaccine injection. And And you can see the number of people increases over time. There's about 100, then about 300, about 3,000 in the phase 3. And then once you submit it for the vaccine approval, we need all the biostatisticians who will then pour over the data, because there's a massive amount of data. Even though the companies analyze, but the FDA has a whole set of biostatisticians will reanalyze the raw data, and that's why it takes time for them to approve. And then once they approve, then it goes to phase 4, and you continue to monitor for a long time. And what happened in the case of SARS-CoV-2, the urgency, the number of deaths is so high, they all apply for emergency approval after phase 3. And that's important because once you have phase 3, you have some idea what's going on. So it's okay to apply to do the emergency use of the vaccine. That's so much for development of vaccine. So I want to get into the virus. So before I get into the virus, I apologize if this is too basic for you, but we need to understand what is DNA, what's RNA, right? We hear about RNA virus. So as you know, we have 23 pairs of chromosome. We get one set from our mother, one set from our father. Most of you would imagine the chromosome to be in an X structure. And you can see that this X structure really, if you stretch it out and stretch it out until you can see what in a fine print, what it is made of. It made up of a bunch of letters, which is A, G, C, T, but it's actually chemicals, and we call them bases. And our human genome is about 3 billion of them, right? And this is called the DNA, or deoxyribonucleic acid. And how long is our DNA? The smallest chromosome is, of course, is the Y chromosome, and it is about 1.53 centimeter. And the largest chromosome is chromosome 1, and it's about 8 centimeters long. And if you take all our 23 pairs and attach them and you stretch them out, they are about 6.5 feet long. It's about 2 meter length is our human chromosome. So it's the largest. I wouldn't say ours is the largest, but it is pretty large. So when talk about genetic material, information that the mother transfers to the child, right? The parents to the child, you know, even a virus to its progenies, or bacteria to its daughter bacteria. The genetic material is mostly DNA, right? The deoxyribonucleic acid. And that information from the DNA has to be somehow transcribed. Somehow you take that information and you need to make the protein required. For example, you need to make your hair. So that hair is a form of protein, even though it's a keratin, right? So how do we go from the gene to the protein? The information from the DNA has to be somehow transcribed into a message. And the message, we call it RNA, right? It's a ribonucleic acid, so it's slightly different from DNA, and it's only single-stranded. And then that gets translated into protein. However, some viruses, the genetic material is RNA. Okay, they don't have the DNA, but just the RNA. A very well-known example is the flu virus, is an RNA genome. And of course, SARS-CoV-2 is also an RNA genome. And compared to the human genome, you can see 3 billion bases versus 29,751 bases is the SARS-CoV-2 genome. And SARS-CoV-2 is the virus that caused the disease. It is a coronavirus. Why is it corona? If you look at these red components, it looks like a crown. If you look under the electron microscope, it looks like a crown as well. And that's why they call it coronavirus. And this coronavirus caused the disease, and we now call that COVID-19. And the common symptoms are dry cough, fatigue, fever, and muscle ache. And of course, there are a lot of uncommon symptoms, and there are severe symptoms. There's a shortness of breath, difficulty of breathing, and coughing up blood, etc. And this is where, when you have the severe symptoms, you better be in a hospital with the oxygen support, and hopefully, you come out of that severe disease. So let's look at the SARS-CoV-2 virus, right? So here's the virus. You already saw that this corona is made up of what we call a spike protein. And if you can imagine, that's what our immune system going to see because it's on the surface. And it's covered with this protein is embedded in this lipid bilayer, which is all membranes. Our membranes are made of lipid bilayer. And this virus has an RNA genome. There is implications of why, what it means to have an RNA genome. We can discuss that later, and a number of other proteins on the surface. Let me draw your attention to this, what causes the corona, the spike protein. This spike protein basically is what actually recognizes or attaches to our host cell. Excuse me. So the host cells has a protein called angiotensin converting enzyme 2 receptor. So the virus can't simply enter any cells. It has to attach to this receptor and then enter the cells. And now we know there's more than one receptors, but for most, it's good enough to know this. This particular receptor is the major receptor that the virus actually attaches, then enters the cell. And most of the vaccines are based on this protein called the spike protein, okay? Because if we can stop this guy's interaction, we can stop the virus from causing us disease. And if you look at the global landscape of the virus, as of December 2020, there are about 184 vaccines under development. All kinds of vaccines. We talked about various kinds earlier, and the numbers are all listed here. And you can see most of the vaccines are protein subunit, nucleic acid-based, or recombinant viral vector. And we'll talk a little bit more when we talk about the SARS-CoV-2 vaccine. And 95 have entered human trials. What phases are they in? There are 36 in phase 1, 25 in phase 2, 20 in phase 3, 7 have a limited use, right? They only covered a phase 2 trial, or they haven't completed the phase 3 trial. And so, but they had applied for emergency use. And we have 3 vaccines approved in this country for full use in many countries, and some are under application. Those are the Mrna, Pfizer BioNTech, and AstraZeneca Oxford vaccine. And then, of course, by now we have already abandoned the 4 of them. And again, we can discuss that later. So if you look at what are the leading vaccines, here are the leading vaccines. The first 2 we talked about, and then the Gamaleya. Gamaleya is a COVID vac, or is Sputnik 5, is the Russian vaccine. And then AstraZeneca is this, has a very long name, and they're also known as AZD1222. Of course, in India, they call it Covishield. And then there is a bunch of Chinese vaccines, CanSino, Sinopharm, Sinovac, Sinopharm Wuhan, and then there's also an Indian vaccine. And some of them in the various phases of trial, there are some emergency use and limited use. So these are the 5 vaccines that Malaysia is actively looking to acquire. What are they? They have signed a contract to get the Pfizer vaccine from about 14 million doses. And then they have signed with Sinovac, CoronaVac, it's called CoronaVac, another 40 million doses. And you can see that they did start a clinical trial in Malaysia, about 3,000 plus subjects. The study is incomplete. And then I have no idea how stable is this. This is what did they inactivate is the whole virus. The information about Sinovac is not available. How about the vector DNA vaccine, which is the AstraZeneca, AZD1222, or the Covishield, as well as Sputnik, and CanSino is also there. And again, Malaysia has signed on to receive 6.4 million doses of AstraZeneca and 6.4 million doses of Sputnik 5. And those in green, the phase 3 is not completed. That means that I don't have access to much information about these vaccines. And it's expected that March is the earliest. Most of the vaccine will arrive in the Malaysian soil. So let's quickly look at these two vaccines that are mRNA vaccines. And I told you earlier, most vaccines are focused on the spike. All the vaccines, most of the vaccines are focused on the spike protein. And in fact, the Pfizer vaccine uses the entire genetic information for the spike protein. The difference with the Moderna is it only uses the small region. This is where the virus binds to the host cell, and they just made 3 copies and put it together to create their vaccine. And we are going to watch a quick video here, and then we'll come back to the talk. And this video is, I really like it. It gives you, can you guys hear it?
It all started with isolation of the virus from an infected patient in China. The coronavirus was sequenced and its genetic information was uploaded to a public database. Working together, scientists at the NIH and Moderna identified the sequence for a key protein on the surface of the virus, called the spike protein, as a good vaccine candidate. The instructions for making the spike protein were then encoded into an instruction molecule called mRNA, which could be administered directly to patients as a vaccine. The vaccine is injected just like other vaccines. The mRNA is taken to immune cells, where it instructs cells to make copies of the spike protein as if the cells had been infected by the coronavirus. Other immune cells, shown here as red and orange figures, are then able to learn about the spike protein and develop ways to protect the person if they ever come into contact with the actual coronavirus. What makes this approach different is that you don't need to make the virus itself to make a vaccine, a time-consuming and intensive process. Instead, you use the information from the virus and administer the information directly to the patient. Essentially, the patient makes their own vaccine. This cuts out the middleman. Using this approach, NIH and Moderna were able to go from sequence to a vaccine ready for human testing in record time, just 42 days.
So I hope you enjoyed watching that video, because it really gives you a quick overview of how that vaccine works. So let me go back. So the Pfizer vaccine is the same as the Moderna vaccine, it's just the component is different. So, and then how about this AstraZeneca vaccine, right? So AstraZeneca vaccine is basically is a DNA vaccine. And what they use is, remember, any vaccine has to be delivered to our body. What is the best way to deliver to the body? In case of the mRNA vaccine, we cover it with the lipid particles, because the lipid particle to resemble our skin. And so it can, when it comes in contact with our skin, it melts into the cell. In the case of SARS-CoV, for vaccines from this AstraZeneca and Sputnik, what they use a virus that causes us a very mild cold. We don't even, we just get a small cold, like a flu-like symptoms, it goes away, right? So it's maybe lasts around 6 to 7 days. So we take that virus, remove the information of the virus, and then introduce just the spike protein gene. So what that allows us is that, remember, the virus is a program to enter your cell. So this virus with the new information will enter the cell, and then we will make the spike protein. And again, you don't have to make the protein and purify the protein, which is much harder. And the nice thing about the virus vaccine is that you can freeze dry them and they'll be fine. And they also are very stable, right? And again, this technology, the people in the field has been working for 20 plus years as to how to make this kind of vaccines. And in fact, Ebola was the first one that was approved, got an emergency approval. And okay, hang on a second, it's refusing to move. Okay, so here is how the virus, imagine this piece of DNA with the information on the spike protein is introduced in this virus we call the adenovirus. And this, when the adenovirus touches the skin and it is get engulfed, right? It's called endocytosis. And then this virus is programmed to go and get closer to the nucleus, where it injects the DNA into it. And then this gets destroyed, the virus head gets destroyed, and then the DNA get then translated into messenger. Remember I told you about the messenger, and then the messenger is then taken out of the nucleus where it's get translated, right? And so the DNA cannot replicate. It doesn't have the information to auto-replicate, so it cannot replicate, right? And so this RNA is then translated to make the protein, and the protein is then expressed on the surface of the cell that acquired the virus. And immediately our immune system says, hey, this cell is infected. We need to destroy these cells. So they will destroy this cell. In the process, they will create antibodies to protect you from the future infection from this virus. So this is in a nutshell how the vectored vaccines or the virus-based vaccines work. And, there's a lot of stories going around about, there are pig cells, people used all kinds of chemicals, which is so untrue. In the case of, I'm just going to focus on Mrna, Pfizer, and AstraZeneca, where the information is given to us, right? So remember, Mrna and Pfizer's RNA vaccine, it clearly has messenger RNA that is synthesized, and then it will covered with lipids and then sugar to stabilize your message. And then you have other chemicals that are traces from the making of the virus, right? And then in AstraZeneca, and of course there is no lipid, but it's an encapsulated inside a virus. And they also list you what are the traces of chemicals that might be in the sample, because remember, you have to purify it. In order to purify, you use all kinds of chemicals to clean it up. And that's the list of chemicals there. And I don't see any human blood or human products or animal products here. And then in terms of vaccine, of course, we want a stable vaccine. And unfortunately, Pfizer vaccine requires a very low temperature, minus 70 degrees. It doesn't last very long in the refrigerator. It's only up to 5 days. Whereas AstraZeneca and Sputnik and CanSino, they are all virus-based, and they all can last. We only now have data up to 6 months, 2 to 8 degrees. Even the CanSino, I don't have any data. I can predict that it can also last at 2 to 8 degrees, because all three virus vaccines are based on the same virus. The delivery, of course, whether you deliver as a lipid nanoparticle or viral particle in themselves, and you do intramuscular injections. Mostly we do 2 doses, and their days are different for different vaccines. And you can see Pfizer's 22, AstraZeneca 28. Now they're recommending 3 months. Second day later, and Sputnik is 21. CoronaVac is right now, the studies are just 28 days. And CanSino, it looks like you need 3 doses in order to get a good vaccine response. And the phase 1, I won't go into in detail. Basically, you try different amounts of the vaccine, and then you settle for one at phase 3. Pfizer combined 1 and 2, and then 2 and 3, and they settled for 30 micrograms. And then in AstraZeneca is the most confused studies, but they wanted to use just 5 times 10 to 10 vaccines, that is 5,000 million vaccine. And what happened is they discovered that if you give half the vaccine for the amount for the first dose, and the second dose come back with twice the amount, the immune response was much better. But they tested both combinations. And also, remember, I talked about placebo. You must give, and the placebo chosen by all these companies do not make sense to most of us. They use saline here, and in AstraZeneca, they used the meningococcal vaccine. Maybe this is the way to give people meningococcal vaccine, which is much needed to prevent pneumonia. And then Sputnik here, all I want you to focus here is that number of sites that were tested, and you can see Asia is not figured in, so we don't really know how well Asians will respond to this vaccine. And you can see that some South American countries, in Africa, only South Africa was included. Sputnik was only tested in Russia. CoronaVac and CanSino, the first 1, 2, and 3 were all in China. It's a completely Chinese population, but there are some ongoing vaccine trials in different countries now. The other important thing to recognize the fact that they have to have a male and female more or less equal number, because men and women respond differently to vaccine, right? Our body makeup is different. And most of the studies do a pretty good job of, matching male and female in both in vaccine group and placebo group. And then the other important thing to notice is that Pfizer had the age range of 12 to 89. And then if you look at Sputnik, the age range was 18 to 60, and then AstraZeneca was 18 to 69. Of course, this is the virus. And interestingly, what I noticed is that Sputnik has more males than females. And in fact, AstraZeneca had more females than males, right? So how do we respond as a female? And then here is a bunch of measures, that they all differ in number of days. They follow the patient and what they look at. So this is not very critical, but basically we know what are the things. The major things to note is that whether there is an immune response to these vaccines, whether there were any adverse reactions, and whether there any side effects. They have to report that. And if you look at the side effects, and most of the side effects are the injection site pain, swelling, and redness. Sometimes you feel tired or fatigued, or headache, muscle pain, chills, joint pain, nausea, swollen lymph nodes, vomiting, and fever, and flu-like symptoms. These 3 vaccines ask the patients, did they have any one of these reactions, right? They followed. Sputnik only focused on whether you had a headache or fever. If they asked for something else, I don't know. They did not report that. And we don't know what is CoronaVac and CanSino are tracking. And also, even though many of them do that, they don't report. And I want to focus on these 2 guys. When you look at the number of people that were tested, given vaccine and placebo, and how many of them actually got any reaction, you can see Moderna is less than 10% reaction. And even people who got placebo, no vaccine had reaction, right? Sometimes to some extent, it's also psychological. And with the Pfizer, we had about 30% reaction. And this is the vaccine some of you might get. And most of them are general or injection site reaction. But if you compare to people who didn't get reactions to number of people who got reaction, more than 80% did not have any reactions to Pfizer vaccine. Sputnik, they did not report how many people had any reaction or not. The other thing they look at when they look at whether the vaccine is good or not is how many people in the 2 groups get COVID-19, right? So if you look at the vaccine group, there was only 1 case of severe COVID in this Pfizer, and you can see these are number of COVID positive. In the placebo group, the number increased over time, right? And then there were many, many cases of severe COVID-19 cases in the placebo group. And so overwhelmingly, the vaccine group was protected. And it's the same thing with the Moderna, and I don't have exactly where the severe cases. In the Sputnik, they also report the same graph. If you have a vaccine, you have less chances of getting a COVID occurrence. Whereas if you take placebo, there were a number of cases of COVID plus based on your DNA testing. AstraZeneca has a similar graph, but they present it very strangely. All I can tell you is that there were 10 severe COVID cases in the control group, not the vaccine group. So all in all, the vaccine does protect people once they get the vaccine. But what is important, I told you, all these vaccines were done in the various countries. And you can see here, the majority of the people are whites. And in some of the vaccines, we have some African Americans, some Hispanics, and then this is Native American Indians. So there's some of them. We as Asians, we want to know what happened to the group of Asians, right? And you can see there's 1,300 or 1,300 in Moderna, slightly more in the Pfizer, very little in AstraZeneca. So let me summarize that. Let's look at here. So these are the 4 vaccines, and you can see the whites were majority of them, the whites, 82 to 98%, and the efficacy is between 90 to 94% in various these vaccines. How about Asians? In the case of Moderna, we got 100%, even though the population tested were very small, 4.7%. Unfortunately, it's not, Malaysia doesn't have a agreement with Moderna for their vaccine. Pfizer, we got only 74.4 vaccine, percentage efficacy. That means that 3 in 10 people will not have any protection. And then in AstraZeneca, had much smaller number. They did not report this. And then Sputnik, of course, did not have any Asians. That number will change shortly. So that is all about the SARS-CoV-2 vaccine. And, you already know, and I don't have to convince you that getting a vaccine protects people, right? And we know getting an annual flu vaccination can prevent deaths. And I hope all of you will get the annual flu vaccination. It can, this is our US studies, and they say that you can prevent 35 million illnesses, right? And look at that, about 34,000 can be prevented from dying. And this, if you want to get an idea what is 34,000, a small town in Ipoh, like Ipoh Garden, probably have 34,000 people. And imagine all of them dying at one time because of a flu. And so you really need to think about and getting a flu vaccination. But what is more important with the SARS-CoV-2 vaccine? There are people becoming poor. There's people who don't have food on their table. They can't go to work. They can't make money. And people, some of us are lucky. We have a job that still pays us to work from home. Many people can't do that. So you have, you're pushing people to poverty, and we're going to have a lot more poverty to deal with because of SARS-CoV-2, because of COVID-19. And then with all that issues, there are people who are very hesitant to take vaccine, okay? And I'm hopefully you guys, all of you here today, will become an advocate for vaccine. And many diseases can be prevented, and you can ask why there are enough vaccines for all the diseases. To develop a vaccine, it costs more than $1 billion. And as I told you, it takes more than 10 years. And then it's not really interest for the pharmaceutical companies. I rather give you daily medication, daily give you paracetamol so that you feel better, or should I want to cure you and then I don't get to sell my drug, right? So it's not of interest. So what changed with COVID-19? This is what changed. Also, the vaccines are usually for diseases that attack poor countries and people in tropical countries. But if you look at the COVID-19 burden, Europe was hard hit, US was hard hit, okay? And Malaysia is right here as well, right? So then there was an incentive for the countries like the US putting in a lot of money into this development of the vaccine. They almost put in $3 to $5 billion in the vaccine development. And so people say, I don't want to take this vaccine because it's rushed. It was not rushed. I hope you're convinced. And then they say, oh, the body can cope without vaccine. Sure, you can die from it. It's a choice you make, right? And the vaccine will give you COVID-19. The question is, none of this, we are not giving you live vaccine to cause a disease. So it can't give you COVID-19, right? And then there are people, literally people they interviewed in UK, they say, oh, the vaccine has a chip that people will track you. That is completely insane to think about that. And then the other things, oh, it will tweak your DNA. No, this DNA cannot integrate, they cannot replicate, or the RNA cannot even enter the nucleus, so they all will be disintegrated after certain time point. Like the RNA vaccine literally has only 5 minutes of lifespan, right? Within 5 minutes, the RNA is disintegrated. And then the side effects are worse than COVID-19. I'm not sure about that. Are you rather have your severe COVID with the breathing difficulties and stay in a hospital, or you have little pain on your site of injection, right? And then make women sterile. How? Convince me how it can do that. I have no idea. And then this age-old argument that vaccine causes autism. It has been debunked over and over again. There was one group that started it, and constantly people come back and say it's cause autism and diabetes. You know, you eat a lot of sugar to get diabetes, right? You eat, you know, your ice kacang and all your drinks that are full of sugar. That's what causes diabetes, not the vaccine. And I'm sure some of you have more, you know, myths, and I would love to debunk most of them if I can. So the vaccine facts, it's safe and effective. It can prevent deadly illness. And imagine you can't even be next to the person dying. And this is what happened in China. They just covered the body and dumped in the truck. And then they dig mass grave. And this is in New York, right? It would provide better immunity. And if there's no vaccine and if you don't get vaccinated, that will continue. And you can't be close to your loved ones. And the choice is really yours. And so you really need to think about the efficacy versus the risk, right? And for me, it makes no sense. You know, the associated risks are too low as compared to dying from it or getting very sick from it. I'd rather take the vaccine. And with that, I will stop and take questions.
Isn't it fascinating, friends? Isn't it absolutely fascinating? Right, friends. So I'm going to ask, because there's just too many people in the group, I would be grateful if you have questions to type on the chat box, and I'll then channel it to Kalai. I think it's much easier. Otherwise, you know, it will come from all over the place. I do have a first question though, just to keep the ball rolling, kick the ball rolling, and that is late. I think just what early because I read somewhere that is it the AstraZeneca vaccination is no longer given to a certain group, age group and above, 65 and above or something. So is it that they have found things, found reactions that that are adverse? You're on mute, dear. You're on mute. So we haven't heard.
Yeah. So so when they didn't see those reactions in the at the clinical trial, and I remember the number of people that were above 65 is rather low in the clinical trial. And then when they decided that the people above 65 should be the first group of patients that should be injected, and then they had some adverse reactions for people who had pre-existing hypersensitivity reaction, right? Those people who carry around the epinephrine needles to prevent any reactions. So those people were reacting. And so what they do is they said, if you have pre-existing sensitivity to anything at all, and you are the one who's running around with epinephrine needle, you should not take this vaccine, at least AstraZeneca, right? And that is the reason why people with the pre-existing conditions, they have to watch them for 15 minutes to make sure they don't react, because it's stoppable, right? You can immediately stop with epinephrine needle to prevent the reaction. But if you're a healthy, you know, you're fine, they observe you for 30 minutes before sending you home, right? So that you don't have any adverse reaction. So it's only seen in AstraZeneca for 65 plus with adverse reaction. Yeah.
Yeah. Okay. Second question before I turn to the chat box that I see some questions coming in is that, you know, for those who have been infected already, you know, COVID positive patients, does it mean that, because I heard somewhere, and it could be hearsay and it could be completely false, that they are immune for a period? Now, is that true?
It is true. What they recommend is that so far we know people with COVID infection, they are still recommended to take vaccine, but they are asked to wait for 90 days before they go for the vaccine. So at least now we know that up to 90 days, they don't get reinfected. People do get reinfected after 90 days. Okay, so you really basically 3 months, right? And that was also a lot of people who was not sick to get the vaccine to protect themselves. So it is true. You are recommended to wait up to 90 days before getting the, if you're already infected, right? You have recovered.
Oh, fascinating that even virus work in cycles of 3 months.
Yeah, but the thing is, it's not cycles of 3 months yet, it's how far we have come. We have to keep monitoring them over. So the time frame we have just information on that is 3 months. Maybe if we continue to monitor those people, maybe it's a 4 months, it'll be 6 months.
Sure.
So for now, we have only data up to 3 months.
Sure. Okay. So Sulaiman asks, are all vaccines with this has the same effect? I think you discussed this a little bit just now.
No, the different vaccine, all the vaccines are supposed to generate the neutralizing antibody, right? So the final goal is to protect you from the viral infection. So that final effect is the same. But how quickly they generate and what they use to generate is slightly the action. And as you saw, the efficacy of each vaccine is different, as I pointed out. And Mrna has 100% in Asian, Pfizer is 75% in Asians, and then we don't know about the others. However, even within the whole study, it's 90% plus. And then I didn't have a chance to tell you that CanSino that is now currently tested in Brazil is only 50%. That means only 1 in 2 people will get protection. So now they're recommending 3 doses, and maybe in another 2 months, we know that with 3 doses, we're going to end up protecting more people, but jury is still out on that.
Okay. All right. So I'm going to merge 2 questions because they more or less are on the same topic, and that is, there are multiple COVID variants that are now, developing, right? They're evolving. So how would the current vaccine, when you take the current vaccine, how are you being protected against all these variations or mutations?
Okay, good question.
The same thing again, yeah.
So it's a very good question. So first of all, remember I told you this virus genome is RNA, right? Our cells are made of DNA. So the virus, in order to replicate, in order to make more viruses, it has to bring its own enzyme to make the virus particles. And this enzyme is not very good at proofreading. What is proofreading? If in our case, when our DNA in our baby, in our fetus, as it divides and replicates and make more cells to create the baby, if they're not faithful, you will have 1 finger less or you will have 1 year less. So it has to be 100% faithful, right? Whereas in the case of virus, that enzyme is not very faithful. It doesn't know how to be faithful. So it's going to continuously create mutations that will create variants. And the idea is these variants to also is a sort of evolving into stabilizing their lifespan. So any pathogen will try to establish an equilibrium in order to survive with the human. There's no reason to kill us, because if they kill all the humans, they cannot replicate any further, right? So they have to establish a stability between the human infections and their survival. So these changes or the mutations will continue to come, just like flu virus, it continues to evolve, right? Because it's the same RNA. So as far as do these vaccines work on them, the answer is yes and no. Yes, in the lab, we see that the vaccines, because remember, you can test it in the lab very quickly, whether this antibody actually binds to that particular virus, and it just takes us, less than 2 days to test. So the test shows that most of these vaccines are able to neutralize that virus in the lab. Whether it neutralizes in the human, we don't know yet, but obviously as we give the vaccine, we also will get samples from patients
from coming,
from infecting. Yeah. So I hope that answers the question.
Yeah, just a sec, huh? Right. Yeah, I think that's beautifully answered. There are more questions. Let me pull them up, Kalai. So there are several questions asked around the same thing. So I will ask you, there are various of this. Now, so I think there are concerns about, let's say we have some underlying illness already. Can we take the vaccine? So one is, I'm going to just pull it all together. One is, let me see, one is rheumatoid arthritis. The other is prostate cancer. The other is osteoarthritic patients, colorectal cancer. And let me see. Yeah, so basically, if you have these type of underlying illnesses, whether it's cancer, whether it's arthritis, are you able to take such vaccines?
So first of all, I want to answer the cancer. Any diseases that makes you immunocompromised, right? So in the cancer, your immune system is compromised. There are other diseases where you don't make the antibody in order to protect yourself. So those cases, it's recommended you get vaccinated, right? Even though we did not test patients with immune compromised patients in the clinical trial, but people with diseases that the immune system is compromised, like transplant patients and cancer patients, should take the vaccine. So in terms of arthritis and all that stuff, we didn't study that population per se, but this vaccine should not affect, because, for example, many of the arthritis, it's autoimmune arthritis, that is due to our immune system attacking our DNA, right? So it's not attacking the viral DNA. So I would say the only group that we have data for is there were cardiovascular patients, there were some obese patients, and diabetes patients were involved in many of these studies, and it was fine. Those people had no reaction, or rather the study doesn't have contraindicate, right? There's no contraindication for these vaccines. So that's my short answer. But if you're a very specific population, most of like, for example, if you're a transplant patient or if you're a cancer patient, if you go to the cancer website, especially the US run Cancer Association, they have a specific instructions for you about COVID-19 vaccine. So if you have rheumatoid arthritis, go to the rheumatoid arthritis patient society run websites. Remember, don't go and look at any sites. Okay, there are a lot of fake news out there in the internet. You need to go to a website that is run by the society, right? Like the Cancer Society or a transplant society. There they have instructions given to you. If you're a transplant patient, what is your recommendation? So don't take my word for it, go check that.
Okay, excellent, thank you. So someone asked, how long is the lifespan of a vaccine? In other words, let's say we take the first and the booster, how long do you think it'll last?
So as I told you earlier, we have only data up to 6 months, right? Or 5 months, right? So far, the vaccine is lasting. Those who have got positive response, the vaccine is there. So not the vaccine, is the immune response is there. But in terms of how long the vaccine lasts in your body, literally 5 minutes for the RNA to last, and then the protein is made, and in 2 weeks, you made the antibodies and you get a second vaccine, the antibody is made, and about 20 plus days, it comes down and it stays, the memory cells are there. So what you're looking at is how long does the memory last, right? We hope that we all have memory, we don't get dementia. It turns out the immune cells do get dementia. But as of now, for the SARS-CoV-2 vaccine, the trial started in February, in March, and June. So we only have data until now, and and these patients will be continued to monitor for next 10 years to see how long they last. And if you talk about measles, mumps, and rubella vaccine, we know people above 55, they're losing the memory for those vaccines, and you should get vaccine. So another plug I'll give you. People find that people have a good childhood vaccination program. Countries with good childhood vaccination program seems to have a little bit more protection. And it turns out that it's potentially that measles, mumps, rubella vaccines, MMR vaccine seem to protect the individuals. So go get your MMR vaccine. I just got mine, okay? Go get your MMR vaccine even before you want to wait for your SARS-CoV vaccine to give you a pre-protection.
Did I hear you right? You said MMR vaccinations seem to be offering some protection.
Correct.
Oh, fascinating. All
And
right.
all of us, most of us here are above 50. Should go get it.
I just love the way you simplify everything, and it's so easy for us to understand, Kalai.
Thank you.
So there's another fascinating question. It says, what do you think? Let's say if I took from Moderna, then my booster I take from another Pfizer or something. Is it possible that we take a different type of one and the second?
So you depends on the vaccine, right? So Moderna, all of them target the same protein. It should create the similar, but there is no studies combining, even though they are looking at it, whether if you can go to Moderna and get Pfizer, it should be fine. From a scientific point of view, I can tell you that taking a second vaccine that is different, except for not about the inactivated vaccine, right? Because inactivated vaccine is the whole virus. So we don't know what is our immune system is reacting to. But in the case of Moderna, in the case of Pfizer, in the case of AstraZeneca, they all use the same protein. So our immune system has seen the same protein. And that is the vaccine, probably they will react similarly. But it may, again, we haven't tested that, but it's scientifically, it should be good.
So someone asked, let's say we have taken both doses. How do we know we are truly protected? Do we need to go and take a test to confirm?
So that's what they do, right? Once they give the vaccine, they continue to do the test to see if the virus is in their system. And they find that in some cases, only the highest were in the cases where there's no placebo, right? And it's possible, like today, I'm suddenly, I met somebody who has coughed onto me, and then I have the virus, right? I'm now infected. It might take me in a couple of hours or maybe a day for my immune system to say, hey, stop, you're not going to cause me disease, right? But during the time, it doesn't stop you from transmitting to people who doesn't have the vaccine. So that's why they say, even after vaccination, continue to mask yourself, right? Because if you happen to get the virus, you have to give time for your body to react. And until then, you could be transmitting the virus to people who are susceptible to the infection.
Makes perfect sense, yeah. So along with that, there is one question that says, after taking a second dose, will it be safe to travel soon after? So I think what you have just answered, it's to that, to that question.
So
Next question is, Kalai, doctors come to you, if they come to you, just a sec, if they come to you and they say, all right, Kalai, we're going to give you a vaccination now. Which vaccination do you want? What would you say?
I want to answer your first question is, if you get the second vaccination, are you ready to travel? Not for 2 weeks, right? Now they tell you that for one is Moderna, they say 14 days. Also Pfizer, they say wait for 14 days before you travel. So your immune system is really ready and ready to fight back any infection. So after second vaccination, wait for 14 days minimum before you start traveling. Plan after your second vaccine. And remember the efficacy, right? If you take a Pfizer, chances are 30% of you won't have vaccination, right? Won't have protection. So you can test it. Of course, you can test whether you have protection. They just have to take your blood and test whether you have this neutralizing protein, these antibodies that will neutralize. So you can certainly ask your doctor to test, can you test if I have the neutralizing antibodies? Then you know whether you have protection or not. So there's 30% of you potentially will not have a protection. And again, it was not tested when they say Asian, right? Did they test in Chinese? Did they test in Indians? Did they test the Thais or the Vietnamese? We don't have that breakdown. We all are different community with a different genetic makeup. We're going to respond differently, right? So Indians will be a little bit ahead because India will test all the Indians there. And then the Chinese a little bit ahead because China will test all the Chinese there. How about the in-between groups, right? So we have to be mindful. And what was the second question?
Yeah, so the second, if you have a choice, which would you choose?
So first of all, you guys have no choice, right? When they call you, say, Lei Chin, you have to come and get your vaccine today. Just go. Whatever you get, right? But if I have to choose at this point, I will go for Moderna. Moderna has the best efficacy. And fortunately, in the US, I might get Moderna. So among Asians, at the point, we only have some data for AstraZeneca. We don't have any data for Sputnik, right? We don't have any data for CanSino yet, right? And like I told you, CoronaVac, we don't have any data. CanSino is only 50% effective in the Brazilian population. We don't know about the Chinese and population. So, stay tuned. They do publish the phase 3 trial. And in fact, I just told you the Sputnik phase 3 trial, literally data came out 2 days ago. So you have to stay tuned as to what population they use, how it's protected. So it's the best is if you have a lab, if you guys want to be entrepreneurial, start a serology lab that will test people who got vaccinated and test their antibodies in 2-3 weeks to see if they have a positive response or negative response.
Okay, tell us a little bit about herd immunity. We keep hearing this phrase. And does it work, herd immunity?
It does work, but you have to get about 80 to 100% of the population, at least up to 80 to 90% of the population immunized. Then the 20% will be protected. Because when you give it to them, right? But the thing is, they can't give it back to you because you're protected. So the herd immunity does work, and it needs to have, depending on the vaccine, it anywhere between 16 to 80% or 90% of the population immunized for herd immunity. And some vaccines do better than the others.
Okay. Now, this thing about bottling of vaccines in Malaysia, what does that mean? And will it be affected in any way, the efficacy, the quality?
Okay, so imagine if you get 1 liter of vaccine, right? In the case of this inactivated vaccine, right? You can get 1 liter of the virus. And then in order to put it in small bottles, they can aliquot it. They can put it in small little bottles. If the company, I know Pharma Gia or something, I believe that is, yeah.
Pharma Niaga.
Pharma Niaga, that's correct. Pharma Niaga has agreed to want to pack these vaccines in Malaysia, as long as they use the clean system, right? They have the right clean system, and they qualify for it should not affect, because like I told you, these virus particles, they're extremely stable at refrigeration temperature. Many of these pharmaceutical companies have a very cold rooms to do all this work, and it should not affect unless, this person who is working is careless, and, then something could happen, because contamination has happened in the past, even in the best cases, right? So, and then this person who is packaging should not also be tested. They are negative for the virus, because you can accidentally introduce the virus into the vaccine vials if you're not careful. So the staff has to be tested constantly, on a daily basis before they start doing work. So we hope that Malaysian companies will take all the precautions necessary.
Okay. And I will pick this up as a final question, because it's almost, let's see, what time is it there now? What time is it there, Kalai?
Oh, it's 9 a.m. and I'm good to go for another 2 hours. So as long as people can ask all the questions and make sure I've answered most of them.
Okay, one question is interesting, and I will ask it anyway. So since Asians are smaller in size, will the dosage be smaller too?
No. And a quick answer is no.
Okay, I suspect that was the answer. Now, another person is, you did recommend taking MMR just as a protection if you have no access to a vaccination, right? So this person says, I took it when I was young. Do I need another jab? I think your answer would be yes, right?
Yeah. We all remember I was telling you there's a dementia, the memory cells are lost. So I took it when I was young, and in fact, I even took it 20 years ago, and I decided to get a booster last month, right? So it doesn't hurt you, you have the slight sore on your arm, but it really creates fresh memory cells, so your system is much more robust. So I would say, all of you, go get your MMR vaccine.
I think there's also someone who asked this question, and I think this is basically what it is. With so few vaccination doses available, and of course, these are going to go to the more vulnerable, those who are in the front line and all that. The rest of us will probably have very little access, and so therefore, during that intervening period, maybe getting the MMR vaccination is a level of protection, right, that it provides.
Yes.
Yeah.
Absolutely.
Okay.
And you're getting yours tomorrow, right?
Okay. So another comment is about aspirin allergy. That adverse, something about aspirin. Are you?
What about aspirin?
It says yeah. How about, I don't understand this question. How about aspirin
Sorry,
allergy?
may I speak? Sorry, I'm so sorry.
Sure.
I'm the one. May I?
Of course.
Okay, thank you. Thank you. Sorry, I put the question very early on, so I didn't see that you picked it up, and then when I typed it again, wasn't clear. All right. You see, I have a severe aspirin allergy, so I go into anaphylactic shock, right? And in the beginning, I think it was a few months ago, there were some data released that if you have a severe allergy to medication or even certain food substances, right, that there will be an adverse reaction when you get vaccinated. So I wanted to ask about this. I mean, do you, Dr. Kalai, have any further data on this? Thank you.
So I discussed that in the lecture, and I say that if you have any serious adverse reaction, you are recommended not to take, or if your doctor recommends you should take because you have other underlying reasons to take the vaccine, you should have your epinephrine needle next to you, and the doctors has to observe you 15 minutes to see if there is any reaction. Is taking aspirin as any contraindication with the vaccine? I have no idea. But I would say the recommendation in general is if you have serious allergic reactions, you should hold off from taking vaccination, hold off from taking any vaccines.
Okay, makes sense. Yeah, I think always consult a doctor, a professional, right?
Yes, and I'm not, I forgot to say, I'm not a medical doctor, I am a PhD.
PhD doctor. Yeah, so there are a bit of comments here and there, but basically I think you have addressed all of them. And yeah, and friends, I just because people are already dropping off, and I do want to happily, yeah. So if you want to thank Professor Kala, can you just type in your comments onto the chat box, because I will be sending her all the entire transcript. So this is she wakes up particularly early to do this for us, and she speaks to us from so far away. This is a rare treat. So if you wish to thank her, please type in the chat box. But just one announcement, friends. Next Friday is Chinese New Year, so there's no session. All right, even though we are all celebrating at home, there is no session. And the following weekend, following Friday is something that will be of interest to quite a few of you. It is on Bujang Valley. If you're familiar with Bujang Valley, Bujang Valley is a little location up in Sungai Petani. The professor who is working a lot in that space will be talking to us about how that is an early civilization, not very different from Angkor Wat. This is our Angkor Wat in Malaysia. So we're going to have it the week following the Chinese New Year week. All right. So with that, friends, I'm going to ask everyone to unmute ourselves. But meanwhile, as you unmute yourself, I just want to say, Carla, you were more than brilliant. More than brilliant. You made everything so easy for us all to understand a very complex topic.
And controversial.
And controversial topic. And you just made it so beautifully simple. At the same time, you didn't spare us the science. You shared the science with us, which is brilliant. You know, I'm so proud to have you as a friend, Kalai. So can we all clap our hands and thank Kalai? Unmute and help me thank Kalai.
Thank you. Thank you, Yemi. First of all, we in the US, we always ask permission before recording. So please, we did record this lecture. Just for information, I want to say thanks, Yemi, for bringing me here today. And I'm very happy that I could impart some information to our Malaysian families and friends, because I think we need to do more of this session. And as I was telling Yatmi, you know, I spoke to 800 people in India, and this is the first time I'm talking to a large group of Malaysians. And with that, I also want to wish everybody Kong Hee Fat Choi. I really miss being home for Chinese New Year, and have a great holidays. And thank you all. Thank you for being here today.
Thank you, doctor.
Thank you. Thank you.
Thank you.
Thank you, Professor Dr. Kalai. And love to your dear husband, Giri. Bye.
Thank you.
Everyone.
Bye.
Hope to
And
see you
Happy
again.
New Year to everyone.
Thank you.
Thanks, everybody.
Thank you.
It looks like my sister logged in too. Yeah.
Which is your sister?
That's Kalaivani.
Oh, where is Kalaiwani staying, living?
She's in Penang.
Oh,
Ani, show your face. She has the frost. Vani? Okay.
She signed in very early because I saw that. I thought, no, I know your name is not that, so I didn't admit.
That's me. That's me.
Hi.
That's
Hi.
me. Hi.
Vani is, she was penolong pengarah at the nursing college in Penang.
Yeah.
Oh.
Matajam, and then she moved to Ulu Kinta, where they started the university for the nurse. And she just retired 2 months ago.
And I am doing part-time lecturing in Fatima Nursing College, Ipoh.
Oh, fantastic. Your parents would be so proud of both of you. So proud of both of you. Amazing. I'm so proud of these Ipoh girls here.
Yes.
Yeah.
Go Ipoh. Hey, go Buntong, you know, I'm from Buntong. Not just Ipoh, Buntong Kaki.
Yeah, that's true.
Yeah.
Just to want to clear, I'm afraid that people, whether I've got it wrong in understanding, MMR does not cover or does not give immunity for COVID-19. Am I right?
It protects. It doesn't give you full immunity.
Just
it protects. Yeah. You know, it's a they find that people in countries where there is a strong, so there is what we call in scientific, there's a molecular mimicry, right? Is somehow the antibodies generated for MMR seems to recognize the virus, right? It's not 100%, but it seems to recognize. So it's called the molecular mimicry. And so people looked at what is that antibody generated by that vaccine is recognizing in the virus. So they find that MMR has the closest mimicry in terms of giving protection. So, people the scientists who really believe this protection exists, because many early studies showed that why doesn't Indians get severe COVID, right? Why India has so many infected yet there is not much. Why certain countries in South American countries we don't get. And if you look at their vaccine policies, their vaccine schedule, and then they say that this particular vaccine seems to have the highest chance of providing protection. And it is scientifically validated information.
Thank you.
Interesting.
Thanks, sister.
This is like watching, the movie, when the movie has ended, and then while the credits are running, you hear more information about the story. This is what it is.
The uncut version.
Yeah, the uncut version. So those who are still
Exactly. Movies are made 10 hours and then they cut. And,
Yeah.
and then they release the uncut versions later, or, find new parts of the uncut versions of useful
But,
data.
I have to tell you, you see that beautiful piece of art behind Kala? It's her drawing.
Wow.
So I say, oh, ye of many talents.
That's one of my favorite for COVID artwork.
COVID artwork.
I call it, I titled it a celebration.
Yeah, it
Very
gives you that,
positive.
it gives you the explosion of colors, right? So it's a celebration. So tell you what, I think most have signed off. So Kalai, such a pleasure, really, such a pleasure connecting this way. I might call on you again, maybe in a few months' time, if there's more developments. Would that be all right?
Oh, certainly. As soon as we get more data on those Chinese vaccines, and we can certainly revisit some of these. And I don't have to give all the backgrounds unless people want it. And then we can go into what is the data for Chinese vaccine is.
Right.
At the moment, it don't look very promising, but but Malaysia has signed on to bring so many of them, right?
Yeah.
And then so it was very interesting too, is that the myths, a lot of the myths generated with the first two vaccine is sort of dying down, And so it's a bit good, we can start with the Chinese vaccine and then discuss the myths again one more time. It's always best to reinforce the information.
Yeah.
So we are not very clear where the vaccines are going to come from. We understand it's Chinese, but is it going to be a mixture or a palette of different vaccines that Malaysia is going to bring on board, or would you know?
So you missed. Oh, I'm still screen sharing.
Sorry.
No.
You just
Wait. Give me 1 second.
Sure.
You missed this.
Sorry.
No, it's okay.
I also read something.
No need to apologize among friends.
I also read that the Russian Sputnik vaccine has got very good efficacy.
So did you see this? Can you see my slide?
Yeah.
So here are the vaccines that Malaysia has signed the contract or agreed to receive. So they're getting 14 million doses from Pfizer. And then Sinovac, they have also signed for 14 million doses. And then this is where the Pharma has a contract, I believe. And then this AstraZeneca and Sputnik also, they are sending 6.4 million doses each. And I was telling earlier that even though so all these vaccines have no information on how well, except Pfizer, which was 1,700 patients, we know about 74% effective. All the other vaccines, we don't know how effective will it be with the Indians or with the Malaysians or Chinese or Malays. We have no idea. We might get some information as soon as we get the India trial is done, how the Indians might respond as soon as the China releases their phase 3 trial. They just literally this week released the phase 2 trial for one of the Chinese vaccine, a Sinovac, I believe. So CoronaVac, they just released it. So anyways, we don't have phase 3 trial data for any of the Chinese vaccine. And that's actually good once they finish. Yeah?
And there's no Moderna vaccine for Malaysia. Maybe I should try to order from eBay or maybe to get you some. They need to send some.
The other thing with the Pfizer, right? This is what I'm very concerned, and I know I've seen it happen, and my sister can attest to it. The Pfizer vaccine, you need a -70 freezer to keep it freezed. And it only lasts few hours in the refrigerator. And many nurses in Malaysia, they are very, very poorly trained. And they leave it around on the bench. And by the time you get it, you may not even have the right stuff. You may getting just junk in your blood, in your system. And that is, sorry, but my sister will agree with you. I was privy to how poor training the nurses have. And when I'm sick, I want my sister around to bully them.
Wow.
You can't do this. No, I'm this. She has the right test. When she was working, do spot check in hospitals. She'll go spot check the nurses and how they do.
Can I adopt your sister as well?
She's very adaptable. You can adopt her.
I think that's you are spot on, right, Kalai? I think when there's more info on the Sputnik, particularly when there's more info on the Chinese vaccinations, because we are probably getting more from from China. Yeah. So I think our friends would appreciate understanding the science behind it, which is safe, which is not safe, the efficacy. We would appreciate hearing from you again. So I think not very long from now, we'll probably be calling on you again soon.
No problem. But as soon as I will send you an information as soon as the phase 3 trial comes out.
Perfect, because I think you are watching this space closer than I would, so I would appreciate hearing from you. All right. So how can we thank you, Kai? How do we thank you? If you are here, I will give you a huge hug. You've just been hugged.
Social distance cannot hug.
Quick hug, a tight hug. And with lots of love, Yetmi. It's always a pleasure to see you. And nice to see you, Venu. Venu, you did well.
Can you please tell your husband that for me too?
He's hearing. He's listening.
Okay, Kiri, you did fabulous then. You did fabulous then.
Yeah, he's laughing there. No, we both did well, Yetmi. We picked a good husbands.
All right, pleasure, Kedah. And the two Kalai, such a pleasure catching up with you too, Kalamani. All right, take care, friends. Yeah, have a good day.
I'm signing out. Okay?
Have a good weekend. Bye.